Disease. Diseases can be broadly grouped into infectious and non-infectious. Classification of diseases: infectious diseases, non-infectious diseases, infectious, contagious, non-contagious, non-infectious, cancerous, deficiency disease, allergic diseases, and addiction. Infectious communicable diseases. Diseases which are easily transmitted from one person to another are called infectious diseases. Infectious diseases are very common and every one of us suffer from these at least sometime or other. Some of the infectious diseases are like AIDS and are fatal. Infectious diseases are of two types. Contagious disease. These diseases are transmitted by contact between two persons. Example: measles, chickenpox, smallpox, ringworms, conjunctivitis. Non-contagious diseases. These diseases are transmitted to vectors. Example: malaria, filaria, dengue, AIDS, etc. Non-infectious disease, non-communicable diseases. These diseases do not transmitted from infected person to healthy person. Example: cancer, diabetes, hypertension, and kwashiorkor. Among non-infectious diseases, cancer is a major cause of death. Study of cause of disease is called etiology. Irrational fear of disease is called pathophobia. A wide range of organisms like viruses, bacteria, fungi, protozoans, helminthes, etc. Could cause disease in man. Such diseases causing organisms are called pathogens. Parasites cause harm to the host by living in them, or therefore most of the parasites are pathogens. The pathogens can enter our body by various means, multiply and interfere with normal vital activities, resulting in morphological and functional damage. Pathogens have to adapt life within the environment of the host. For example, the pathogens that enter the gut must know a way of surviving in stomach at low pH and resisting to various digestive enzymes. Cancer. Cancer is characterized by an abnormal and uncontrolled division of cells to form a tumor or neoplasms. So, cancer is a cellular disease. Oncology or carcinology is the study of cancers. Cancers causing cells are called oncogenic cells. Cancer is one of the most dreaded diseases of human beings and is a major cause of death all over the globe. More than 1 million Indians suffer from cancer, and a large number of them die from it annually. The mechanisms that underlie the development of cancer or oncogenic transformation of cells, its treatment and control have been some of the most intense areas of research in biology and medicine. Cell growth and differentiation is highly controlled and regulated in cancer cells. Its breakdown of these regulatory mechanisms, contact inhibition property shown by normal cells, by virtue of which contact with other cells inhibits their uncontrolled growth. Cancer cells appears to have lost the property of contact inhibition. As a result of this, cancerous cells just continue to divide, giving rise to masses of cells called tumor. Loss of cell adherence and cell differentiation is called anaplasia. Tumors are of two types: benign and malignant. Benign tumors normally remain confined to their original location and do not spread to other parts of the body and cause little damage. Malignant tumors are a mass of proliferating cells called neoplastic or tumor cells. Neoplastic cells grow very rapidly, invading and damaging the surrounding normal tissues. As their cells actively divide and grow, they also starve the normal cells by competing for vital nutrients. Cells slaughtered from such tumors reach destined sites through blood, and whatever they get lost into, they start a new tumor there. This property is called metastasis, is the most feared property of malignant tumors. Causes of cancer. Transformation of normal cells into cancerous neoplastic cells may be induced by physical, chemical, or biological agents. Agents which cause cancers are called carcinogens. Radiation. Ionizing radiations, X-ray and gamma rays. Non-ionizing radiations. UV rays causes damage to DNA leading to neoplastic transformation. RBE, relative biological effectiveness, refers to damage caused by radiation. Nuclear leakage is carcinogenic. Physical agents. Tobacco chewing causes oral cancer. Chemical carcinogens. Tobacco smoke causes lung cancer. Lung cancer is also caused by chromium fluoride. Biological carcinogens. Cancer-causing viruses are called oncogenic viruses, have genes called viral oncogenes. Oncogenes were discovered by Bishop and Warmus, Nobel Prize 1989. Epstein-Barr virus causes Burkitt's lymphoma. Human papillomavirus, HPV, causes cervical cancer. Herpes virus 8 causes Kaposi sarcoma. Kaposi sarcoma is marker for AIDS. Hepatitis B virus, HBV, causes hepatocellular carcinoma, liver cancer. Several genes called cellular oncogenes or proto-oncogenes have identified in normal cells, which when activated under certain conditions could lead to oncogenic transformation of normal cells. Cancer is caused due to activation of proto-oncogene to oncogene and inactivation of tumor suppressor gene. Tumor promoter is a substance that activates transcription in cancer cells. Types of cancer: carcinomas. These cancers are located in epithelial tissue, ectoderm or endoderm. Cervical cancer, breast cancer, brain cancer, lung cancer, skin cancer, stomach cancer, etc. Cancers of gland is called adenoma or adenocarcinoma. Cancer of melanocytes of skin is called melanoma. Sarcomas. These cancers are located in connective tissues and muscular tissue derived from mesoderm. Sarcomas include the cancer of bones, muscles, adipose tissues, cartilages, and lymph nodes, etc. Cancer of adipose tissue are called lipomas. Cancers of bone is called osteoma. Cancers of lymphatic system or lymph nodes or spleen are called lymphomas. Hodgkin's disease is a type of lymphoma characterized by enlargement of lymph nodes and spleen. Blood cancer, leukemia. Abnormal increase in WBC count of blood due to increased formation in blood marrow is called leukemia. Cancer detection and diagnosis. Early detection of cancer is essential as it allows the disease to be treated successfully in many cases. Cancer detection is based on biopsy and histopathological studies of the tissue and blood and bone marrow test for increased cell counts in the case of leukemias. Biopsy: a piece of suspected tissue cut into thin section is stained and examined under a microscope. Histopathological studies by a pathologist. Techniques like radiography, use of X-rays, CT, computed tomography, and MRI, magnetic resonance imaging, are very useful to detect cancers of internal organs. Computed tomography uses X-rays to generate a three-dimensional image of internal objects of an object. MRI uses strong magnetic fields and non-ionizing radiations. MRI is most accurate and safest method to accurately detect pathological and physiological changes in the living tissue. PAPS test is used to detect cervical cancer. Antibodies against cancer-specific antigens are also used for detection of certain cancers. Techniques of molecular biology apply to detect genes in persons which have inherited susceptibility to certain cancers. Identification of such genes which predispose an individual to certain cancers may be very helpful in prevention of cancers. Such individuals may be advised to avoid exposure to particular carcinogens to which they are susceptible. Example: tobacco smoke in case of lung cancer. Acid phosphatase is a marker enzyme in diagnosis of prostate cancer. Acid phosphatase, prostate cancer. Treatments of cancer. Common methods are surgery, radiotherapy, chemotherapy, and immunotherapy. Surgery. In surgery, the whole cancerous tissue is removed. Radiotherapy. Tumor cells are irradiated lethally, taking proper care of surrounding normal tissues. Cancer cells are damaged by radiation, while others are not due to the cancer cells are immature and undergoing rapid division. I-131 is used for detection and treatment of thyroid cancer. Chemotherapy. Several chemotherapeutic drugs are used to kill cancerous cells. Some of these are specific for particular tumors. A weed called Catharanthus roseus Satabahar provides two anti-cancer drugs, vincristine and vinblastine, which are used against leukemias. Majority of drugs have side effects like hair loss and anemia, etc. Most cancers are treated by combination of surgery, radiotherapy, and chemotherapy. Immunotherapy. Tumor cells have been shown to avoid detection and destruction by immune system. Therefore, patients are given substances called biological response modifiers such as alpha interferon, which activates the immune system and helps in destroying the tumor. Immunomodulators can stimulate or inhibit the immune system immunity. Every day we are exposed to large number of infectious agents, but only a few of these exposures results in disease. This is due to the fact that the body is able to defend itself from most of these foreign agents. Overall ability of host to fight the disease-causing organisms conferred by immune system is called immunity. Immunity is of two types: innate immunity and acquired immunity. Innate immunity, it is non-specific type of defense. Innate immunity is present at time of birth. Innate immunity provides different types of barriers to the entry of foreign agents into our body. Innate immunity consists of four types of barriers: physical barriers, skin or a body is main barrier which physical barrier. Skin on our body is the main barrier which prevents entry of microorganisms, pathogens. Mucous coating of the epithelial lining, the respiratory, urogenital, and gastrointestinal tracts also help in trapping microbes entering our body. Physiological barriers: acid in stomach, saliva in mouth, and tears from eyes all prevents microbial growth. Perspiration contains lysozyme enzyme that kills bacteria. Lysozymes found in saliva, tears, and perspiration destroy certain bacteria. Cellular barriers: leukocytes, WBC in the blood like PMNL, polymorphonuclear leukocytes, neutrophils, monocytes, and natural killers. Types of lymphocytes, as well as macrophages in tissues, can phagocytose and destroy microbes involved in body defense. PNML provides innate immunity. Innate immunity is provided by T cells. Migration of phagocytes and leukocytes towards the place of infection occurs to diapedesis. Macrophages are derived from monocytes. Macrophages are known as phagocytes. Phagocytes provide non-specific type of defense. Cytokinin barriers. Interferon is chemically protein. Interferon is secreted by virus-infected cells, which protects non-infected cells from further viral infection. Interferon is antiviral substance. Interferon were discovered by Isaac and Lindman. Note: first line of defense of body is skin surface barrier. Second line of defense is phagocytic cells or competent cell system and interferons. Third line of defense is immune system. Acquired immunity. Acquired immunity is pathogen specific. Innate immunity is non-specific. Acquired immunity is characterized by primary response. When body encounters a pathogen for the first time, produce a response called primary response. Primary response is of low intensity. Secondary response or anamnestic response. When body encounters the same pathogen subsequently, then body elicits secondary response. Secondary response is highly intensified. This is due to the fact that our body has memory of the first encounter. Memory cells are formed from T lymphocytes. Primary and secondary immune response carried out with the help of two special type of lymphocytes present in our blood, that is B lymphocytes and T lymphocytes. B lymphocytes or plasma cells produce an army of proteins in response to pathogens. These proteins are called antibodies or immunoglobulin. Gamma globulins are synthesized in lymph and lymph nodes. Antibodies are the compound formed in an organism for inhibiting the growth of another organisms. Antibodies or gamma globulins or complex glycoproteins. If a doctor suspects major deficiency of antibodies in a person, then for confirmatory evidence he looks serum globulins. Segments of an antigen that are recognized by antibody are called epitopes. Study of interaction of antigen and antibody in the blood is called serology. Antigen are found on the cell surface. Antigen is a molecule that produces an immune response and form antibody. Recognition and digestion by phagocytosis due to coated surface of antibodies is called opsonization. Conversion of antigen into harmless insoluble matter by antibody is called agglutination.